to listen to your seminar. I
Ah,
always enjoy your seminar.
well, thanks for coming, and hopefully there'll be some maybe a few new interesting things in there. But thank you.
Well, that'd be exciting. I always like your seminar, like your science.
Thank you very much.
All right.
So James, I saw you keep posting paper in BioArchive. That's really,
Yeah,
really productive. So you,
it's been a busy year. Yeah.
always.
Yeah, I think
I
a
see,
lot
I see.
of projects coming together, which is nice. So,
That's
uh,
great.
and then it's the next challenge, getting from the preprint to publication. But yeah,
Yeah, yeah, yeah. I saw a couple of them were already being accepted, right? So it's just
yeah,
in
yeah,
process.
yeah.
The CLCC1 story,
Yes,
that's
yes.
exciting.
Yeah,
Almost
yeah,
there.
yeah, yep. This one's
Mm.
accepted, so should be out in the next few months.
Oh,
Yeah, really
that's
happy
great.
to
That's
see that
great.
one come together. That was a labor of love, but a lot of work in that one.
I see, I see. I still remember the first time I hear the story in the 2023 ASBMB meeting, right? It was really exciting. Yeah, yeah,
Thank you,
yeah.
thank
It's
you.
great. It's great to see it came out. So,
Yeah, it's been really fun to see kind of, I mean, there are a lot of surprises with that particular
mm,
project. You know, a lot of unexpected directions. And then I think since we put this out as a preprint together with Xiaowei
yeah,
in 2024, there have been several other preprints kind of coming out, seeing similar findings and building on this, which has been really nice to see. So it makes me feel really good.
yeah, 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 yeah.
Yeah, I saw Xiaowei put another preprint on bioarchive. It is about torsins. Did
Yes,
you did you
yeah.
see that?
Yeah, very interesting. Yeah, and
Yeah.
it's his preprint. There's also one in a Drosophila model on torsins, also on bioarchive. Very interesting. Yeah, and they have kind of similar conclusions there. So I
Did
think
you did you
that's a
find
really exciting
torsins in
direction.
your list or?
We hadn't directly looked in that way. We've done a little bit with torsins. Certainly, they phenocopy, but we hadn't gone as deep as what they've done in terms of functional characterization. Um,
Okay.
but it makes a lot of sense. I think there's still kind of a lot of mechanistic questions there to be teased out, but the relationship is quite clear. And even if you just do, there's some of these databases looking using AlphaFold predicted interactions. If you look at CLCC1, what comes up is torsin in those.
Right.
So,
Right.
so I think that's pretty strong, kind of complementary evidence there.
Is Xiaowei coming to your talk today?
I don't know. Is he
Should
here?
be here.
It would be good to have him to be the panelist, one of the panelists.
Yeah, I guess he probably knows too well for the story.
Well, I think he will show up. He's a avid uh,
Yeah.
participant in
Yeah,
many meetings.
that looks great.
Well, let's see. Shall we? Yeah, there. I saw Xiaowei's name.
Oh,
Hello.
okay.
Hello, everyone.
Oh, Chuma is here. I
Hi.
also
Hey,
saw Ning
Xiaowei.
is here. Ning
I
Wu.
see Xiaowei waving. We were just talking about you.
Xiaowei, you are muted. Xiaowei, you muted yourself.
Oh, okay. Sorry. Yeah. Hi, James.
Hi, nice to see you.
Good to see you. Yeah, yeah. Thank you for sending the holiday greetings. Yeah,
Ah, yeah, yeah.
I did not reply. I thought that you may be traveling and replying emails. Yeah.
Oh, thank you, thank you.
So
Yeah.
James,
So you,
would you like to start sharing your PowerPoint?
Yeah,
Let's just
sure.
give a quick test.
Make sure that this is in here.
Yeah,
How's it
yeah.
looking? Success?
Looks great.
Okay.
Oh,
Useful.
Yeah, this is like one of those experiments you can do at home, just mixing kind of paint. This is water soluble paint and oils. Not for me, but I have done this kind of with my kids at home, and kind of nice demonstration of the separation of a lot of the lipid properties, right, of oil and water.
So this
Yeah,
is
yeah.
the most impressive cover page that I've ever seen. Very interesting.
It's all downhill from here, so.
James, you are gonna be at the FASEB meeting, right? Lipid droplet and phospholipid.
Yes, you'll be there?
Yeah, I most likely I'll be there if I can get to enter the US.
Oh,
Please. I hope so. That would be great. That's a really fun one because those two meetings kind of are happening at the same time. So, around 300 or so lipid enthusiasts. So that's a really nice meeting.
Yeah, absolutely. Yeah. 2 years ago we gave back-to-back talks, right? So
Yes.
I think that worked really well. Yeah.
Yeah.
This time I think there are several guys, no, several colleagues from China will come to the meeting. Yeah.
Great. Yeah, I guess 2 years ago that was in Minnesota, and then this one will be in Arizona. So it'll be really hot, but uh, I've been to that location once before, and my goal was to either be in the conference room in air conditioning or in the pool. So Arizona in the summer is very hot, but it'll be fun.
Absolutely. Yeah. I see you in Arizona.
Yes, I look forward to it.
Mhm.
Okay, well, it's almost time. We have a lot of audience still coming in. We can briefly start now. So, hello everyone. Welcome to the Metabolic Edge Talk Series 251. Today, we are honored to have Dr. James Osman joining us. Dr. Osman is a professor and the Doris H. Calloway Chair in the Department of Molecular and Cell Biology and in Nutritional Science and Toxicology at the University of California, Berkeley. He received his PhD from Emory University and completed postdoctoral training at Stanford University. Dr. Osman investigates the mechanisms that govern organelle and lipid homeostasis in health and disease, with a focus on lipid droplet biology and ferroptosis. His work has uncovered the fundamental mechanisms linking lipid storage, lipid peroxidation, and protein quality control to cell fat decisions under metabolic stress. Dr. Osman's scientific contributions have been recognized by numerous honors and multiple distinguished lectureships at UC Berkeley and MIT. Dr. Osman has published extensively in Cell, Nature, and Science. He also actively organized international conferences in lipid metabolism and cell biology. His topic today is mechanisms of cell lipid homeostasis, from lipid droplets to ferroptosis. So we will have four panelists who will join in the discussion after the talk. Now, Dr. Shaobing Zhang from Capital Normal University, Dr. Bo Zhong from Wuhan University, Dr. Zhipeng Li from University of Florida, and Dr. Ling Wu from Huazhong University of Science and Technology. So meanwhile, our audience can post your questions in the chat. We will call you after the panel discussion. Okay, Dr. Osman, we are very excited to hear your story. The stage is yours now.
Thank you very much. And yeah, thank you to the organizers and to the panelists and to everyone attending for the interest. It's a real pleasure to have the opportunity to join you today. Good morning to those in China, good afternoon and good evening to those over here in the US. It's my pleasure to have this opportunity to share with you some of our recent findings and perspectives around the cell biology of lipid homeostasis, with a focus here today on droplets and ferroptosis. So my lab is very interested in the cell biology of lipid metabolism. We're interested in ER-derived neutral lipid storage organelles called lipid droplets, how these organelles form, how proteins target to these organelles, how these proteins regulate lipid droplets, and how they're at times degraded by the ubiquitin proteasome system to remodel the proteome of the droplet, how these organelles interact with other organelles, for example, forming membrane contact sites for the communication of lipids to maintain cellular lipid homeostasis. And we're very interested in the role of these organelles in regulating lipotoxicity, and more broadly in the mechanisms that cells use to defend themselves against the accumulation of toxic lipid species, for example, oxidatively damaged lipids in ferroptosis. And more broadly, we're interested in how these processes go awry in disease states and how we might begin to therapeutically intervene by targeting these pathways and mechanisms as we begin to gain a deep understanding of the biology. So I've structured this talk into two sections. In the first section, I'll focus on how lipid quality in cells is maintained. So this will focus on ferroptosis and mainly on a protein we discovered previously called FSP1 and some of the mechanisms, regulation, and cellular roles of this protein. And then the second portion, I'll talk about some of our work on lipid storage and neutral lipid flux in the ER. So our cells have evolved highly sophisticated quality control systems to combat the accumulation of damaged biomolecules. And this is conceptually shown here that we can have macromolecules that are sensitive to damage. These damaged products can at times be toxic to the cells, so we need quality control systems to ensure that these don't accumulate and cause cytotoxicity. And these can be classified in a few different ways. We can think of some of these quality control mechanisms being preemptive quality control, so suppressing the damage. We can think of some of these as being damage repair quality control, so reversing the damage or repairing the damage. And in many cases, that damaged product is simply degraded or removed from the cell or metabolized into a benign product. And I think a really good example of the quality control systems comes from extensive studies over the past several decades around protein quality control. We know that proteins can misfold, these can potentially aggregate and be toxic, but we have things like chaperones that can suppress misfolding or refold proteins. We have things like the ubiquitin proteasome system or lysosome autophagy pathways for degradation. And we also have homeostatic stress responses, like the unfolded protein response. But what about lipids? Can we think about lipids perhaps in a similar conceptual framework, that perhaps they also can be damaged and be toxic, and perhaps they also require quality control machinery? And I think there are many examples of this now. For example, with polyunsaturated fatty acids, these are prone to oxidation, and this can lead to cell death. And our lab and many others are interested in the quality control processes for these lipids. So how do we combat the accumulation of these oxidized lipids? And this question around lipid quality control led us to become very interested in a form of cell death known as ferroptosis. This is a regulated type of cell death, distinct from other types of cell death like apoptosis. And for us, this is really a type of cell death that is due to the failure in lipid quality control. And ferroptosis is intrinsically tied to lipid biology and lipid metabolism. So polyunsaturated fatty acids, as I mentioned, are prone to oxidation. These are incorporated into membrane phospholipids. In an iron-dependent manner, you have the production of radicals or reactive oxygen species that can lead to the production of lipid proxy radicals and lipid peroxides in the membrane. And if these accumulate to high levels in the membrane, eventually you can get rupture of the membrane, which we refer to as ferroptosis in this form of death. Now, there's a well-known quality control system that combats ferroptosis. This is mediated by the glutathione-dependent peroxidase, GPX4. This employs glutathione to convert these lipid peroxides into non-toxic lipid alcohols. And they're useful small molecules. Some of these we use in our study. For example, arastin, which blocks system Xe-. This will result in a depletion of glutathione, triggering ferroptosis in some cell types, and other molecules like RSL3, which are direct inhibitors of GPX4, which we can also use to trigger ferroptosis in some cell types. And this field is really exploded in recent years due to the potential therapeutic relevance and importance of ferroptosis. With most of the field focusing here on cancer therapy, the idea that perhaps we could trigger ferroptosis as a way to selectively kill drug-resistant cancers. And there's been a lot of data in preclinical models suggesting that this might be a potential therapeutic route forward, though this remains to be translated into actual therapeutics. There's also the idea that on the flip side, that perhaps we could inhibit ferroptosis, and this might be beneficial in ameliorating some of the degeneration associated with degenerative conditions like ischemic tissue damage, neurodegeneration, and others. And so when we began thinking about this problem of lipid quality control and ferroptosis, we noted that in many cancers, inhibition of GPX4 triggers ferroptosis, but in many cancers, they're quite resistant to GPX4 inhibition as well. So they're kind of subsets. And in some cancers, for example, we could delete GPX4 and these cells would continue to grow in vitro and divide, suggesting that there must be additional protective mechanisms beyond GPX4 that are quite powerful to suppress lipid peroxidation and ferroptosis. And so this is the question that really drove this former postdoc in the lab, Carol Brazuker, to answer this. And he wanted to ask this question around, are there additional protective mechanisms that promote lipid quality control and compensate for the loss of GPX4? And to do this, we took a genetic approach, and this was an experiment actually conducted by Zhipeng Li in collaboration with Carel to perform these genetic screens in the lab to look for ferroptosis resistance factors that act parallel to GPX4. And so what Zhipeng did was he took these resistant ferroptosis lines, an osteosarcoma line U2OS, infected with an sgRNA library from our colleague Mike Bassick down at Stanford, and then we simply treat with our GPX4 inhibitor. Now, if we've knocked out a gene that's required for protection, these would die and be depleted from the final population, which we could deconvolve by deep sequencing. And I actually remember when we did this experiment, we were imagining that we'd get many different hits, and how would we deconvolve this, and how would we prioritize? But it became very clear when we actually did the screen what we should focus on. We had one very strong hit with a high confidence score, and this was a gene formerly known as AFM2 that we renamed FSP1, or ferroptosis suppressor protein 1. And this was simultaneously discovered by Marcus Conrad's lab in a companion paper. And very nicely, while we were doing a synthetic lethal deletion screen, Marcus' lab did an overexpression suppressor screen, and we both came to the same protein and very similar mechanisms. So that was a really nice convergence there. Now, just a little bit more data from this paper before I move on to more recent studies. We show that FSP1 knockout, as you might anticipate, sensitizes to the induction of ferroptosis with RSL3. We see classic ferroptotic morphology here. And then we can also do this in dose response analyses. So here is a metric of cell death on the y-axis. As we increase dose, our parental lines are very resistant up to micromolar levels of this small molecule GPX4 inhibitor. But you see our two knockouts are highly sensitized. So here it's almost a binary effect, or at least 2 or 3 magnitude sensitization. So this paper now was published around 6 years ago, so I'd just like to summarize mechanistically what we determined in this paper, and then I'll move on to some of our recent findings around FSP1 and ferroptosis. So as I mentioned, the primary defender of lipid peroxidation and ferroptosis that had been studied was GPX4. In this paper, and in Marcus's companion paper, we discovered a second pathway, FSP1, that seems to operate in parallel to GPX4. And I think what ultimately emerged was mechanistically very interesting. So FSP1, in contrast to GPX4, doesn't convert lipid peroxides into lipid alcohols. Instead, FSP1 is an oxidoreductase, and it takes quinone antioxidants like CoQ, converts them into the reduced radical trap form, and then suppresses the propagation step of these lipid peroxides in the membrane. And this was important because in addition, because CoQ had primarily been studied in the mitochondria and the electron transport chain, but this, I think, finding also solves maybe a long-standing mystery in the field as to what CoQ is doing in non-mitochondrial membranes. We think there it's playing a role in promoting lipid quality, and this specifically with FSP1 acting at the plasma membrane. So this brought up several questions that we've been interested in addressing in the lab over the last few years. One is this concept around subcellular lipid quality control. And I think here we have FSP1 being a very good example of this, acting at the plasma membrane. But you can imagine that different organelles, different types of membranes are going to need their own types of protection. The lysosome, the peroxisome, the ER, the mitochondria, all of these have unique compositions, all exposed to unique metabolism and redox states. And so do we have different types of subcellular lipid quality control systems, and what are their roles? And then, of course, we're very interested in the possibility that therapeutically targeting ferroptosis and targeting FSP1 might be one approach for a cancer treatment. So towards this first concept of subcellular lipid quality control, one area we immediately focused on was the lipid droplet. We've had a long-standing interest in this area, and also we had seen FSP1 concentrating on the surface of lipid droplets. And so this is one more piece of data from that original paper where we had endogenously tagged FSP1. We see really nice localization to lipid droplets, also to plasma membrane ruffles. We also see FSP1 very nicely biochemically fractionating and purifying together with lipid droplet-enriched buoyant fractions. So this raised a kind of logical question here around what it's doing on lipid droplets. And this is work from a current postdoc in the lab, Mike Lange, very talented. He came from Maria Fedorova's lab in Germany and from a lipid analytics background. And he was very interested in this question. Are lipids that are stored in lipid droplets protected from oxidative damage? And the hypothesis being that perhaps FSP1 localizing to the surface might prevent the oxidation of these stored lipids. And so lipid droplets are unique organelles. They have a core of neutral lipids. They're surrounded by a phospholipid monolayer with proteins decorating their surface, FSP1 being one of these. Their most famous role is in energy homeostasis, in the storage of triacylglycerol for energy, but they really function as hubs of fatty acid metabolism, providing lipids that can be used to make new membranes, make lipid signaling molecules, and importantly, by sequestering different types of lipids, they've also been implicated in lipotoxicity. And of course, they're connected to a wide range of disease pathologies. And so in terms of lipotoxicity, there's now emerging data around ferroptosis showing a role of lipid droplets in suppressing ferroptosis, with the idea being that these polyunsaturated fatty acids, rather than being incorporated into membrane phospholipids, can be sequestered in triacylglycerol within the droplet. So they're preventing the flux of those PUFAs into membranes and making them less prone to oxidation. And I think that there's quite compelling data for this model in the field, but this raises a really interesting question. If you have all these oxidation-prone PUFAs in the droplet, what protects them, or are they protected when they're sequestered? And I think this was a major question that drove the study I'll tell you about in the next few slides. So Mike went forward and performed a series of lipidomic experiments, and I won't go through all of these. I'll just touch on a few key experiments, this one being one of them. And basically what he did in this experiment is he took our FSP1 knockout cells, he induced lipid droplets, because these cells have quite low amounts of lipid droplets under basal states, and then he performed untargeted lipidomics and semi-targeted oxidized lipid analyses. And what's shown here are oxidized lipids that accumulate. And what you can see is these oxidized lipids accumulating in our FSP1 knockouts. And this was a really exciting result because what we saw was that was largely accumulating are these oxidized triacylglycerol species shown in kind of yellow and orange. And this suggests that in the absence of FSP1, we're starting to accumulate oxidized neutrolipids, both triacylglycerol and cholesterol esters. And this was in the absence of any external stress. So GPX4 is active. We haven't inhibited it, for example, with RSL3. We've simply knocked out FSP1. So I think this is an interesting example where GPX4 is insufficient to prevent the accumulation of these oxidized lipids. And maybe this is a more unique role for FSP1, which is on the droplet. So this we're seeing by lipidomics. We can also image oxidized lipid accumulation using a fluorescence probe called BODIPI-C11. So this is an indirect reporter of lipid peroxidation. And what you can see is that when we add this FSP1 inhibitor called FSEN1, which I'll come back to, we start to accumulate oxidized C11, shown in this channel. And this colocalizes very nicely with our lipid droplet marker, LipiBlue. So we accumulate this peroxidized signal, and we can block this with radical trapping antioxidants like fer-1, a suppressor of ferroptosis. So we can see this biochemically through lipidomics. We can also see this by imaging. And we can also reconstitute this in reconstituted biochemical formats in vitro. So what Mike did here is he generated artificial lipid droplets. These are similar to liposomes, except in this case we have a phospholipid monolayer with a neutral lipid core. We can then use D10 as a radical initiator to generate lipid peroxides, and then we can follow the peroxidation with BODIPC11, our fluorescence indicator. We can then use this platform to add back FSP1, different substrates, and look at the consequences for lipid peroxidation. So here what I'm showing you is lipid peroxidation on the y-axis over time. And in the black, this is kind of lipid peroxidation happening in the absence of any added protein, just with our artificial droplet. When we add FSP1 together with our substrates NADH and CoQ10, we see this lag phase of lipid peroxidation. And this is indicating suppression by FSP1. And what happens at this inflection point, actually, is that FSP1 has consumed all of the NADH, and so you start to get lipid peroxidation occurring again. If we actually just add more NADH, we extend the lag phase longer and longer. Okay? And then this in an inactive mutant FSP1, there's no effect and no activity. So this was really encouraging, but what we can't tell from this experiment is whether the triacylglycerol is being oxidized or the phospholipids are being oxidized, because this is an indirect reporter. So Mike went forward and did lipidomics again on these preps, and I'll just show you the data here for triacylglycerol. And what you can see here is that we're getting lipid peroxidation of triacylglycerol that accumulates over time, and we have a similar shape here to the curve. We have suppression by FSP1 when it's present here with its substrates. So FSP1 in this biochemically reconstituted format can suppress lipid peroxidation of neutrolipids and phospholipids as long as it has its substrates present. So what are the consequences to cells if you start to accumulate oxidized neutrolipids? And one way we could test this is we can manipulate the composition of our lipid droplets simply by adding different types of fatty acids. So typically, people in the lipid droplet field might add oleate, which is a really robust stimulator of lipid droplets. This is a monounsaturated fatty acid. But we can also add PUFAs like arachidonic acid, which are highly prone to oxidation, to trigger a PUFA-rich triacylglycerol lipid droplet. And then we can inhibit FSP1 and look at what happens. So we can, again, just to characterize this, we can make a really oxidation-prone droplet. We can add an FSP1 inhibitor. We accumulate these oxidized lipids. We can add a radical trap. We can block these. Now, what are the consequences for that? If we trigger lipid droplets by adding increasing amounts of oleate, the monounsaturated fatty acid, knockout of FSP1 really has no effect, and this is very well tolerated. However, we see something quite different if we add arachidonic acid and we trigger a oxidation-sensitive lipid droplet. In this case, we start to see that loss of FSP1 results in death in the presence of this arachidonic acid, right? So we're getting high amounts of PUFA-containing triacylglycerol and death. And just to remind you again, here we're not adding any GPX4 inhibitor, and this is seems to be a really unique role of FSP1 to suppress this type of death. So we can further show that this is dependent on the lipid droplets themselves, so that PUFA is not simply going into phospholipids, for example, and sensitizing the cells. A large amount is going into droplets, just quantified here, and triacylglycerol quantified here. We can block that with DGAT inhibitors, right, and block the PUFA from going into lipid droplets in triacylglycerol. This is just quantified here. So what effect does this have when we look at cell death and ferroptosis if we block droplet formation but still add that arachidonic acid? And what you can see here is that in the presence of arachidonic acid in our knockout, we have high cell death. If we block DGAT inhibitors with DGAT inhibitors, the droplet formation, this completely suppresses. So in order for FSP1 knockout to have this effect, PUFA needs to be incorporated into the droplet, into triacylglycerol. So I think this raises a really important point about the type of lipid and the spatial constraints and importance for ferroptosis. So to summarize what I've told you in those last few slides is that in addition to FSP1 functioning at the plasma membrane to suppress ferroptosis, it also plays roles on lipid droplets. And I think while the majority of the field has focused on phospholipid peroxidation in membranes, this is perhaps the first study, or one of the very first studies, looking at oxidation to stored neutral lipids in lipid droplets. And what we find is that FSP1 is playing a similar role here. It's also reducing CoQ, and CoQ, both in our reconstituted systems and in cells, is suppressing the propagation of lipid peroxide, triacylglycerol, lipid peroxides. And eventually, if this accumulates to high enough levels in the droplet, this can propagate to other membranes and lead to ferroptosis. How this is propagating from the droplet, I think this is still a really open question and maybe a fun question to think about and discuss together. So I think there are kind of two major take-home messages from the study. The first being that I think this is a good example of this concept and idea of compartment-specific lipid quality control. And here we have this mediated by lipid droplet localized FSP1. The second is the idea that ferroptosis can be initiated at multiple locations in cells. And this probably depends on context, right? So there have been papers in the field suggesting that ferroptosis can initiate at the ER. This is from Brent Stockwell. Rafael Rodriguez has shown initiation from the lysosome, others initiation from the mitochondria. And what I've told you here is we have a context in which we have initiation from the droplet, right? And I think that this emphasizes that different contexts, how you induce ferroptosis, the metabolic state of the cell, are going to be important in thinking about this. And of course, these are not mutually exclusive models and likely all take place depending on the context. Okay. So along the way, as we began to perform these studies and see the sensitization to ferroptosis in certain cell types by FSP1 inhibition, we became very interested in the possibility of therapeutically targeting FSP1. And this is work from Joe Hendricks, now a scientist at Vulcan Therapeutics, and a current postdoc, Amalia Magriotti. And this was motivated by many findings in the field. So for example, we had observed that across around 1,000 cancer cell lines, this is data from the Broad DET map, that FSP1 strong expression strongly correlates with cancer cell resistance to GPX4 inhibitors. All of these are GPX4 inhibitors, including RSL3. And then we also knew that FSP1 is amplified in certain cancers. This is taken from a paper from Boyi Gan's lab, and that amplification correlates with poor patient prognosis in these cancers. In addition, there were several papers looking at FSP1 in xenograft models, and two really exciting papers that came out in Nature this past October in 2025, one from Thalis Papagiannakopoulos' lab and one from Jesselyn Ubelacher's lab. And I think that one of the reasons this was so exciting is that, at least in vitro, FSP1 is largely dispensable. You can inhibit it, you can delete it, and these cells don't die unless there's some additional stress, right? Either high PUFAs in the droplet or GPX4 inhibitor. But in vitro, it's dispensable. But in vivo, there's something very different was observed, and that once you start to move into the in vivo context, FSP1 shows the cancer cells show a much stronger dependency for FSP1. And this was shown in a few different models, GEM models, orthotopic xenografts, and others, both with genetics and small molecules, suggesting that there is potential to target FSP1 and have an effect maybe as a single agent therapeutic in vivo in cancer. In addition, this was a collaborative project together with Michael Duponge's lab here at UC Berkeley, led by a really great graduate student in Michael's lab, Jesse Garcia Castillo. And they did something a little bit different than what was done in Thalis and Jesselyn's papers. Rather than knocking out FSP1 in the tumor, what they were doing in their studies was knocking out FSP1 in T cells or Tregs, and looking at the consequence for the immune system. And if you knock out something like GPX4 in T cells, conventional T cells, these all die. But if you knock out FSP1, the T cells survive. And what we're even more excited to see is that knockout of FSP1 in Tregs leads to reduced suppression of conventional T cells, and you get enhanced tumor immune responses. So when you lose FSP1, the gene name being AIFM2, you actually restrict tumor growth. So I think this is really exciting because it suggests there might be synergy between targeting FSP1 in the immune system and in the tumor itself. And this should be out next week in Science Advances. So this really motivated us to start to screen for small molecule inhibitors of FSP1. And this was done together with Julia Schletsky and Eddie Weary here at the UC Berkeley Drug Discovery Center. It was an activity-based screen using pure FSP1 and using CoQ oxidoreductase activity and the consumption of NADH for the screen. We screened 115,000 small molecules, and then the hits from this were triaged and then screened in an orthogonal cell-based assay. And here I'm just highlighting some of the molecules that came out of the screen. You can see that these lumped into a few different classes, these sharing this thiazole ring structure here, this class 1. But we have other groups of FSP1 inhibitors that have completely distinct pharmacophores and chemistry to work with. And we're continuing to do medicinal chemistry and characterize several of these different compounds. And I'll just present a little bit of data on the most potent compound to come out of this screening deck, which we refer to as ferroptosis sensitizer 1 or FSEN1. So this molecule in vitro inhibits FSP1 with a fairly respectable IC50 around 300 nanomolar in this in vitro assay. And we're continuing to do medchem around this to push this number further down. In cells, we can see nice suppression of FSP1 activity. This is an experiment done in KEAP1 mutant lung cancer lines, so they have high NRF2. These are extremely resistant to GPX4, so high amounts of RSL3 have pretty much no effect on these cells, at least in vitro. But you can see if we treat these together with our FSP1 inhibitor, we get very nice induction of cell death, and this is all reversible by ferroptosis inhibitors, as you might expect. So at the end of the study, we had these inhibitors, but we still had a lot of questions around the mechanism of action. And we had also observed that this molecule is selective to human FSP1, and it has little to no effect on mouse FSP1. And so we were a little wondering about what was mediating this species difference as well. And we were fortunate enough to collaborate with Jia at Sichuan University to solve the co-crystal structure of human FSP1 in complex with FSEN1. And this solved a lot of questions for us around the mechanism. So what we saw is that FSEN1 sits right in the substrate binding pocket. So this, based on other structures, is where CoQ would sit. So it seems to be a competitive inhibitor. And it makes these key pi-pi stacking interactions with a phenylalanine within FSP1, phenylalanine 360. And if we mutate this, for example, we lose the inhibition by FSEN1. So it seems to be a very critical interaction for this inhibitor. And if we go and look now at the sequence of human and mouse FSP1, we see that that phenylalanine in the human FSP1 is a leucine in mouse FSP1. So this really explained why the mouse FSP1 isn't inhibited by our molecule. And we can go into the mouse FSP1, mutate it to phenylalanine, and we gain inhibition. So we can humanize the mouse FSP1. And this is actually similar to another FSP1 inhibitor that's out in the field called IFSP1. They seem to bind similar sites and seem to engage also with this F360 residue. So IFSP1 also being human specific. And so I think this study was very useful for us. It solved a lot of questions. It's the first structure of FSP1 together with an inhibitor, provides a mechanism of inhibition, explains the species selectivity, and it gives us a foundation now for rational medicinal chemistry where we can move forward in a more directed, guided manner based on the structure to make changes to this molecule to improve potency. So I want to summarize some of what I've told you so far around the discovery and therapeutic targeting of FSP1. In the first portion, I told you about how we found this protein and became interested in this protein through genetic screens. and the mechanism of FSP1, which utilizes this reduced CoQ to suppress lipid peroxide propagation. We've recently discovered that FSP1 plays roles at intracellular membranes like the lipid droplet, where it prevents the damage to oxidized neutral lipids. Most of our studies to date have been in vitro, but I think something that we're really excited about thinking about and moving towards are physiological models. Where is this going to be important? Fatty liver, MASH, other diseases associated with the accumulation of droplets and reactive oxygen species, I think will be really interesting to look at for this lipid droplet quality control pathway. We've developed small molecule inhibitors of FSP1, several different scaffolds. We've solved the structure of one of these and are moving forward with medicinal chemistry. And one that I don't have time to talk about today, but is out on bioRxiv. This is looking at FSP1 stability. We made fluorescence-based reporters of FSP1 and did a series of genetic screens identifying both transcriptional regulators of FSP1 as well as post-translational regulators of FSP1 stability. And we have an interesting finding around this vitamin B metabolic pathway and the generation of FAD, which leads to post-translational stabilization of FSP1. And this is mostly on bioRxiv if you're interested in this work. So now I'd like to switch gears from thinking about damage to lipids to thinking about how our neutral lipids are stored in lipid droplets and the mechanisms of this. And this, for some of you who may already know the story, will focus a little bit on CLCC1 here. And so just a little bit more mechanism around droplets and how they form. These are ER-derived neutral lipid storage organelles. We have the synthesis of neutral lipids like triacylglycerol, their deposition between the leaflets of the bilayer. At a high enough concentration, these will spontaneously nucleate into a lens, but in cells, this is promoted by a sapin-containing lipid droplet assembly complex, which forms this oligomeric ring structure. And then the lipid droplet emerges from the outer leaflet of the ER into the cytoplasm. And we can have proteins that decorate the surface of the droplet and regulate the functions. And so for most cell types, this is where the story ends. But we have a unique condition in enterocytes and in hepatocytes where the triacylglycerol also has another fate. That triacylglycerol can also be secreted from the cells in the form of lipoproteins like chylomicrons and very low density lipoproteins, VLDLs, right? And these are moving the lipids throughout the cell. And so I think here this highlights something very interesting that I think the field still doesn't understand, which is this decision point. Once you have this triacylglycerol, it's in the hydrophobic phase of a membrane. How does the cell know whether to secrete this or store this, and how is that decision made? And this is going to have big consequences in terms of things like fatty liver, as well as circulating LDLs and cardiovascular disease, that decision point. And so certainly lipid droplets are heavily connected with metabolic diseases, both diseases characterized by low lipid droplets, too few, like lipodystrophies, as well as too many lipid droplets, like fatty liver disease and others. We were motivated in this study around looking at the liver and fatty liver disease, where you accumulate large neutral lipid depots. These are deposits, these are lipid droplets in the liver. And there's been the idea that maybe regulating their deposition could be one approach, therapeutic approach. And so this was one motivation, though we're certainly motivated from a very basic perspective as well to understand just fundamental aspects of lipid biology. How do we store these? How do we channel lipids? How do we form these lipid droplets? And this was a study really spearheaded by a fantastic graduate student, Alyssa Mathewitz, who's now in industry as a scientist. And it's been continued by two current members of the lab, Emily Maimond and Emily Torres, a technician in the lab. And similar to how we approached the problem of lipid quality control, we also approached this using genetic screens, in this case using a FACS-based screen instead of a death-based screen. So here we're using hepatoma lines. These are HA7s. These are similar to HepG2 if you've worked with those cells. They form quite a few droplets. They have properties of hepatocytes, though they're a cancer cell that's cultured in vitro. And these we can mark lipid droplets with Bodipi 493503. This is just some imaging showing this. And then we can use FACS to sort cells that have low or high lipid droplets and identify genes that affect this. And so we performed a genome-wide CRISPR screen, again using Mike Bassick's genome-wide library. Here you can see a much more complex volcano plot emerge in terms of genetic modifiers, as might be anticipated. And many of these jumped out to us and made us feel very good about the quality of the screen. For example, you can see many genes that come out very highly that are involved in triacylglycerol synthesis, acyl-CoA synthetases, desaturases, DGAT2, one of the rate-limiting enzymes in triacylglycerol synthesis. We also saw on the flip side the lipase, PNPLA2 or ATGL, as well as its activator and inhibitor come out. So these made us feel comfortable with the screen. In addition, we saw things like the SREBP pathway and other hits that were emerging. And I think when we were sitting down with Alyssa and trying to think about which of these genes to go after, what was the most interesting, Alyssa was very astute in saying, I think we understand quite a bit about lipid droplets under basal states, but we don't understand lipid droplets under other metabolic conditions. And this motivated Alyssa to go forward and do a number of additional screens. So she did 11 screens under 11 different conditions in duplicate, so another 20 or 22 screens. These weren't genome-wide. This is with a custom synthesized library based on our hits, as well as curated lipid metabolic genes. But I think this was still a big effort from Alyssa, and I think the effort was worthwhile ultimately. And I think here we were motivated for two reasons. One, we imagined that these different conditions could give us genetic modifier snapshots under some different interesting conditions. But also by performing these screens in parallel, we have genetic chemical epistasis where we can start to cluster genes with similar functional profiles. And this will cluster genes with similar potential functions, allowing us to predict functions of maybe interesting new genes and regulators. And this was, to mention, we used some stress conditions like ER stress conditions. We used different starvations, and we used different fatty acid additions. And I won't go through this entire volcano plot here. Ooh, if this goes through. We got the spinning wheel of death here. Oh, there we go. So here we have the volcano plot that emerged from these screens. And I just want to point out at the top, we have what we're referring to as core negative regulators. These pretty much under any condition, when you knock them out, they result in more lipid droplets. At the bottom, we have core positive regulators. That would be things like DGATs, acyl-CoA synthetases. And then we have another in the middle, which I won't go through in detail. We have state-specific regulators. But right at the top, we feel good that at the very top are PNPLA2, the rate-limiting enzyme in lipolysis, and its co-activator, ABHD5 or CGI58, and these cluster together, as you might expect, since they have similar functions. And we have another protein that we became very interested in. This is a protein called CLCC1, which in recent years has started to gain a lot of interest, which I'm happy about. At the time, there wasn't very much known about it. It was known that it was essential in mice and zebrafish. There's polytopic membrane protein. It was a putative chloride channel, but we don't think that it is a chloride channel. It localizes to the ER, the nuclear envelope in Golgi. It's connected with some diseases like ALS, retinitis pigmentosa, and interesting to us is it came up with rare variants associated with altered serum lipids and lipoproteins, which is another reason we prioritize this for characterization. So Alyssa went forward and she made knockouts of CLCC1 again in the H7 cells. And we see a really big effect on the lipid droplet size and morphology. So this is in control cells under basal conditions. If you add this trixin C and acyl-CoA synthetase inhibitor, they'll consume their droplets by lipolysis. And what we can see in the CLCC1 knockout cells is these droplets are much larger, and they're very stable. Even after we've treated with trixin C for 24 hours, they're still around. They seem much larger and resistant to lipolysis. So initially, based on this finding, as well as the clustering with ATGL, we thought this might be a lipase regulator, but it ends up the story being maybe more interesting and a much different mechanism. And so we collaborated here with Anna Paula Arruda at UC Berkeley and Greg Koo at UCSF. We generated a flock CLCC1 knockout mouse to see if what we're looking at in vitro also recapitulated in vivo physiological models. And this was one of the images from this study that I think you can immediately appreciate that these knockouts have profound steatosis, accumulating neutrolipids. They're about 2 times the size. And if you just put this in a buffer, they'll float right to the top. And we had really similar findings to Xiaowei's group, who also is publishing the paper together as a companion paper, back-to-back papers together. This was a really striking finding. I still remember getting this data. I think Emily texted me this. She was in the mouse house at like 11 at night and dissecting these mice, and she texted me this picture. And then I proceeded to gross out my wife and show her all these images. But I was super excited, of course. So we can also confirm this using biochemical mechanisms, lipidomics. I'm just showing you here some of the imaging approaches. So oil red O, as you might anticipate, we accumulate these large deposits. And I think by EM, this is even more striking. You can see in the control liver quite clear. But in our CLCC1 hepatocyte specific knockouts, this is just generated with AAV-Cre, we have these huge droplets accumulating throughout the cytoplasm. And this is with 4 weeks knockout of CLCC1 and no kind of dietary challenge, just under basal conditions, so no high-fat diet or anything. So what's going on here? What's the mechanism? One of the first experiments we did was to just float these droplets and to see what proteins were on them, to see if maybe this would give us a clue to what was changing and what might be altered. And we were really surprised by this experiment. So we floated the lipid droplets, did proteomics, and we can see here is all of these different droplet proteins like perilipin-2, a canonical kind of coat protein or scaffold protein on the droplet, CGI58, ATGL, also known as PMPLA2, FSP1, these were all low. So every droplet protein we looked at were very low in these CLCC1 knockouts. And I think this can be appreciated even more with PLIN2. PLIN2, if it doesn't localize to lipid droplets, it's degraded, and that's what we see here. We see barely detectable PLIN2 after CLCC1 knockout. This can all be rescued by proteasome inhibitors, suggesting that it's being produced, but it's not localizing to the droplet and it's being degraded. And when we look at our droplets, you can see these droplets stained here, the neutral lipid core in blue, but there's no PLIN2 surrounding these droplets. And it wasn't even just that the PLIN2 was kind of light, it was devoid from the surface. So there we were, Alyssa and I were sitting in my office, or what is going on? We have these huge lipid droplets, but no droplet proteins on them. What's happening? And I think the answer became clear when Alyssa moved forward and did some really nice electron microscopy. And what we see here in the control is that we see kind of classic organelle contacts with the lipid droplet contacting an ER here outlined in magenta, just so it's easier to see. But we see something totally different in the CLCC1 knockouts. Here we see, this is a nice image that Alyssa captured because we have an ER sheet here on the left, and one of the bilayers goes all the way around these 3 droplets, and then it connects back to this ER sheet. And what this is telling us is these lipid droplets are not in the cytoplasm, they're actually all in the ER lumen. And this is why we don't have any cytoplasmic proteins on them. This is why they're so resistant to cytoplasmic lipases, because these enzymes and proteins can't access them. They're in the wrong compartment. And just to mention, Xiaowei has very similar findings in agreement with this. So it's nice to see that already we have kind of this validation together. And so we started to think about what this could mean. How could we get droplets all in the ER lumen? And this is a really profound shift in neutral-lipid flux. And we can imagine models in which maybe they're budding off of the inner leaflet for some reason, changes in membrane surface tension. But as mentioned, these are hepatocytes. They can form lipoproteins, and lipoproteins have some similarities with lipid droplets. They're a monolayer structure with a core of neutrolipids. And this gives us some predictions. One is that these are ApoB positive structures. They're also dependent on microsomal triglyceride transfer protein, or MTP. So Alyssa inhibited MTP to look to see the effects. So typically we have cells without Plin2 positive droplets. This is completely reversed if we treat with MTP inhibitor. We get these emerging into the cytoplasm and they're Plin2 positive. These are also ApoB positive structures, and we can also see that there's defects in secretion of these, both in our cellular model, but even more profoundly in the mouse model. This is just quantifying plasma triacylglycerol, but if we quantify ApoB secretion or lipoproteins in the plasma, these are completely flatlined. So a profound effect on secretion. So what is this protein? I mentioned that it's annotated as a chloride channel. We don't think it's a chloride channel. What is it actually doing here? And this is when we reached out to Tim Levine. The only times we had seen similar phenotypes to what we were observing was in the knockout of scramblases with papers from Xiaowei around TMM41B and others around VMP1. And so I emailed Tim and I said, hey, does this look like could be a scramblase? And he said, this doesn't look like a scramblase, but it comes up with a high degree of homology with these yeast proteins. And this was based on protein structural homology searches. And the two yeast proteins are BRL1 and BER6. And we can see the predicted structures here below. And you can see in this domain that they certainly have very similar predicted structures with a long helix extending into the lumen. That's kind of what we're referring to as a knuckle region that's stabilized by a disulfide, a juxtamembrane amphipathic helix, and then at a kind of high degree of angle here, this other helix. And this is very similar in BRL1 and BR6, having this type of predicted structure. So this was really interesting to us. And so what do we know about these yeast proteins, and could perhaps CLCC1 be playing a similar molecular function as these yeast proteins? So these yeast proteins have been studied by several different groups, but this is shown from work from Karsten Weiss's lab at ETH Zurich, in which they've characterized BR1 and BR6 to be involved in nuclear pore insertion, and specifically the fusion of the inner and outer membranes of the nucleus in order to get the pore, a hole in the membrane. You have to fuse this. And this is a unique type of fusion because this is happening within the luminal space here. With the idea that BR1 and BR6, mechanism not totally characterized at this time, are coming together at these intersections and mediating fusion so that you can ultimately have a pore in the membrane. If you lack BR1 or BR6, these are essential in yeast. They're also essential in mouse. You can no longer properly insert nuclear pores, and you end up no longer having fusion, and you have these nuclear blebs or herniations. And this is shown here in these EMs of what this looks like in yeast, these kind of bulges off of the nucleus. And when we went back to our EMs, I feel bad we didn't see this the first time we were so interested in the lipid droplets. But if you look at the nucleus, we actually do see these blebs all along the nuclear membrane here in our Hus7 cells. We also see this in mouse liver in our knockouts, none in the wild type. We can also mark these in primary hepatocytes. This is with a marker from our colleague Christian Schleicher over at Yale, marking these nuclear herniations off of the membrane that accumulate in the CLCC1 knockouts. And since the time where we put out this preprint, there have been several additional preprints. And it's nice to see these validating this finding, making CLCC1 knockouts and seeing very similar phenotypes with the nuclear pore assembly defect. And together with Anna, we went ahead and performed FIBSEM, focused ion beam scanning electron microscopy, to understand at higher resolution and in more detail the ultrastructural changes that are occurring in organelles throughout the liver. And on the left is shown the wild type liver, on the right the knockout. You can certainly see the massive lipid droplets that accumulate in the liver here. As we begin to zoom in, you'll see that in addition to lipid droplets accumulating, there are also changes in ER structure. It looks more fenestrated perhaps, or vesiculated relative to the wild type. But I think the nucleus is one of the most dramatic changes, where you see all of these blebs accumulating around the nucleus. And this alteration and this deformation of the nucleus, this is just a huge lipid droplet deforming and pressing in on the nucleus here, which is quite striking. And since we have serial images, you can go through. These blebs here are actually, for the most part, attached by a membrane neck, as you would expect. And that's consistent with the idea that perhaps we have defects in the fusion step of nuclear pore insertion, and we're getting these blebs accumulating. We can also quantify the nuclear pores themselves to see if indeed we have defects in nuclear pore insertion. And this is shown here from the FibSum images. And this is quantified in this graph. We have a striking decrease in nuclear pores, as well as this huge increase, of course, in the nuclear blebs. And there seemed to be some clustering of the remaining nuclear pores, which we're not quite sure what that means yet, but I think is maybe telling us something interesting. But certainly we're having a profound deficit in nuclear pores and nuclear pore insertion. So to understand maybe a little bit more about the molecular mechanism by how this might mediate fusion, we collaborated with Michael Grabe and at UCSF and his postdoc Nick Van Hilton, and also our continued collaboration with Tim Levine, who was really instrumental to these simulations. And what I'm showing you here is just the predicted oligomer of CLCC1. We've shown that it forms a disulfide dimer in cells, but based on collabfold and other approaches, we think that this forms an oligomer in cells, and that this oligomeric state is probably very important for its function in fusion. What's shown here is Michael and Nick did molecular dynamic simulations in model membranes, and they could make a lipid interaction map. And what that's shown in red here, the interactions with lipids. Very nicely, we see the transmembrane domains come out. That's what's shown here, and that's what we would expect. But we also saw something that maybe we didn't anticipate, and that's this red surface here on the top. And this is an amphipathic helix that extends into the lumen that we think is key and important for this a function and perhaps for membrane fusion. Michael and Nick can also model membrane fusion using this oligomer. So this, the way CLCC1 sits in the membrane is going to bend the membrane, which may be important. And then that amphipathic helix that extends into the lumen, if you put in a second membrane, will insert into that juxtaposed membrane. You get this kind of intermediate state, and then when you morph away the protein, you get full fusion of the two membranes. So I think that this is suggestive. There's also really nice molecular dynamic simulations of BR1 and BR6 from Stefano Vanni and Karsten Weiss with a slightly different model than what we've proposed here, but would also be an idea around how these might mediate fusion. So based on this, we've also performed mutational analyses to mutate some of these key regions, both extending an amphipathic helix, which would disrupt the oligomer state, mutating those disulfide cysteines in the stem loop on the interior. And then I think this one was maybe my most interesting one, which was mutating that amphipathic helix, which we think inserts into the juxtaposed membrane. And just showing the data here for nuclear pores, you can see that wild type completely rescues the phenotype, but these mutants that would be predicted to affect oligomerization or that amphipathic helix are non-functional despite being expressed at normal levels. So I'm going to stop here. I'm going to summarize the two stories I presented today. The first story being really focused on the discovery of FSP1 as an anti-ferroptosis factor that mediates this effect by recycling quinone antioxidants like CoQ, its additional role at intracellular membranes, which I think brings up some very interesting concepts of organelle quality control and how ferroptosis can be initiated from intracellular sites, our progress towards developing small molecule inhibitors of FSP1, and then the second story really just focusing on CLCC1 and this paper that should be out soon, and our thoughts around the mechanism. We think that CLCC1 molecular function is likely that it's playing a role as a membrane fusogenic factor, and this would be the first to be discovered to be fusing membranes facing the lumen. And we think that this is important both at the nucleus, where this is necessary for nuclear pore assembly, but also for lipid droplets and regulating lipoprotein secretion, where we hypothesize that this may play a role in lipid scavenging, though I think that a lot of the mechanisms here remain to be determined. So when we lose CLCC1, we really start to accumulate these giant intracellular lipid droplets and accumulate these large nuclear membrane herniations. So I just want to end by thanking the members of my group, if this will advance. There we go. Oops. And the most important slide. Again. Let me just share this one more time. Computer's struggling. I want to thank the members of my group, while this loads, that I tried to point out along the way, as well as all our collaborators, both at UC Berkeley and abroad, that contributed to these studies. Oh, here we go. Let me see if I can get this acknowledgement slide. Here we go. So both current members and past members of the lab, our collaborators, and all the funding agencies that made this possible. And I'm happy to take any questions. Thank you.
Thank you so much, Dr. Ozma. This is really, really outstanding talks. So this is really, really exciting about like two research directions in your lab. One the FSP1 and also CLCC1. Both are really exciting. I have been heard this story before, but I think there's a lot of new data adding into the story. This is really great. So I think now we're gonna move into our panel discussion. So the first panelist is Xiaobing Zhang from Capital Normal University. Dr. Zhang, would you like to start?
Yes, I heard these two stories before. But today's talk reminds me how interesting these stories are. I actually have two questions. The first question is on your first part of work. We all know that under normal physiological conditions, C20 PUFA, like arachidonic acid, the percentage of arachidonic acid is actually very low. It's probably on the scale of 1 or 2%. So when you add arachidonic acid to the cell culture, presumably this fatty acid has two routes to go, right? One is go to the membrane as phospholipids, the other is go to TAG. And in your ferroptosis assays, how much of that, cell death inducing is from the plasma membrane or microsoma membrane, and how much is from the TAG and lipid droplet?
Yeah, this is a great question. Yeah, we quantified incorporation of the fatty acid into the neutral-lipid and into phospholipids by separating the phospholipids and the neutralipids and then doing GC-FID, quantitative fatty acid profiling on those two pools. And at least in these cell types, and I'm sure there's differences in different cell types, it's primarily going into triacylglycerol. And we know that if we block droplets with DGAT inhibitors, we block the ferroptosis. So at least in this case, it seems to be very dependent on the droplet. And I think that one of the reasons for this is even if it flows into the membrane phospholipids, there we have parallel protective pathways. And so once it goes into the membrane, GPX4 is still active and it can suppress this form. But with the droplet, we think that maybe FSP1 is the dominant player here. And we start to accumulate overwhelming amounts of oxidized neutral-lipid. Ultimately, this will have to progress, though, to the plasma membrane to mediate rupture. And how this kind of propagates, I don't think we really know at this time.
Okay, thank you. My second question is on CLCC part. So you propose two functions for CLCC1. One is in the fusion of ER membranes or nuclear pore membranes. The other is for the formation of intra-ER superlipid droplets, if I understand correctly.
Yes.
So are the two functions or two aspects functionally connected? Are they causing or enhancing or sensitizing each other? Is there
Yeah.
an explanation?
No, that's a great question. I think that we're able to uncouple this in one direction. For example, we know that if we block MTP, this will correct the cytoplasmic emergence of the droplets, and this does not correct the nuclear pore defect. So I think we can uncouple in that direction and say that at least we know that the nuclear pore defect is not due to this alteration in droplets. It's hard to correct the opposite direction. But what I can say is that the nuclear pore defect seems to be very general, whereas the lipid defect and this large luminal lipid droplets is specific to hepatocytes, cells that form VLDLs. So if we knock out CLCC1 in like HepG2s, we see the same effect on the droplets. But if we knock out CLCC1 in UTOS or other non-hepatocyte cells, the lipid droplets all emerge fine, but we still have the nuclear pore defect. So I think that evolutionarily, the nuclear pore defect and its role in nuclear pore assembly is probably its ancient function all the way down to early organisms. And it gained this function in liver, maybe in enterocytes, that we don't know this, for regulating the lipid droplets in the internal luminal lipid secretion.
Okay, great work. Thank you for your answers.
Thank you.
Thank you.
Okay, thanks, Dr. Zhang. So next, we will invite Dr. Bo Zhong from Wuhan University. So Dr. Zhong, please.
Yeah, thank you, Xu. James, can you hear me?
Yes.
Yeah.
Okay, yeah. James, very good, very nice talk. I have 2 or 3 questions about your first part. As we know, there are in addition to TAG, there are also cholesterol in the lipid droplets. I was wondering whether FSP1 also affected the metabolism of cholesterol in the lipid droplet or in the cytoplasm. So in your lipidomic data, did you see some data on the cholesterol part?
Yeah, I can't say that we've seen anything specifically around cholesterol itself, though we see cholesterol esters, and these can also be oxidized. So we accumulate both oxidized triacylglycerol and oxidized cholesterol esters. And cholesterol esters, I think the impact there on the lipid peroxidation and the neutral lipids is that could be a really interesting topic. There's some in vitro data suggesting that cholesterol esters can kind of suppress lipid peroxidation, at least in artificial droplets. But what this means, I'm not sure. I think on the cholesterol ester side, I'm really interested in conditions where there are very high cholesterol esters, in which you get this liquid crystalline phase separation and these rings of cholesterol esters, and what this might mean for oxidation. And this might be important for foamy macrophages, for example.
Okay. So following that question, do you think FSP1 can directly, reduce oxidated TAG? Or do you think a loss of FSP1 results in TAG release from the lipid droplet, and then the released TAG will be metabolized to oxidated phospholipid? So what do you think?
Yeah, I think for the first question, does FSP1 directly reduce triacylglycerol peroxides? So I don't think it enzymatically reduces these in the, for example, turning these into alcohols like GPX4 would, or some of these peroxidases would. More that it's preventing proxy radicals from propagating, mediated by vitamin E or CoQ as the hydrogen donor